When an infant is introduced to solid foods, this is also during a period of time where their milk feeding also decreases. And this is accompanied by adaptations in both the gastrointestinal tract and the immune system. So when a baby's born, they have quite a low diversity of those bacteria. And as they're introduced to breast milk and then subsequently to foods, that diversity changes. And we don't really know in terms of the diversity what is the appropriate or optimal diversity of that bacteria to optimise health. So again, our research is trying to really explore the different types of kind of prebiotic foods that can be given to infants during the introduction of solid foods that may enhance the bacterial diversity and also optimise their immune function. And they were given that for 6 months, which they could basically have in their diet as a complementary food along with other foods as well. Over that 6-month period, they came to the University of Auckland for visits where we collected information from the parents about the health of the infant, what the infant had been eating, and also a number of biological samples, so fecal samples, urine samples, saliva samples, and also breast milk samples from the mothers. We're going to again feed them the kumara for 6 months, and we're going to collect again a number of biological samples so that we can analyse the microbiome diversity in the infants over the 6-month period. There's a lot of synergies, a lot of collaboration across the programmes, and that really provides that additionality of science excellence across the high-value nutrition, which is a really important aspect of the National Science Challenges. In our pilot study of the 40 infants, when they were around 9 months of age, they had had between them 54 episodes of upper respiratory tract infections, so we know it's very common. And so we're trying to understand whether by altering the microbiome diversity, whether there's some crosstalk with the immune system and whether we can try and benefit or optimise the microbiome and therefore optimise the immune system, and by doing that reduce the number of upper respiratory tract infections in that time. None of this happens in isolation. It's not all about me, it's about the people that I work with and collaborate with.